record. All right, so now we're recording, share, slides. And we are going down and there. All right, back to it. Thank you all. All right, so we have a filter. We trap bacteria. We can tell what we had by taking that filter, and then we can put it on something like a Petri dish with growth media and see if anything grows on there. If it grows, something was trapped in your filter. You could also do this, one second, with your sterile liquid. So if you take a sample from your liquid and put it on a plate and something grows, that shows you your filter was not very effective. Yeah, what's up?
So this is like the same process some people use like charcoal and water?
Yeah, exactly. So like the charcoal filters that we use to filter our tap water, this is going to act similarly. I don't know if charcoal is going to be reactive and grab molecules a certain way or another, but either way, you're going to trap stuff as it tries to pass through the carbon. Yeah. And that's why you can only use it for so long. Eventually, all the trapped stuff is going to start to go into your water, so that's why you replace your filters every so often. In a lab setting, especially in something like a cadaver lab, you're going to end up, or a surgical room, a maternity ward, we're going to have high efficiency particulate air filters, HEPA filters. This is going to get all the small stuff out of the air. We're going to keep air cycling. So in a lab setting, you're always going to have airflow continuously going. You don't want stagnant air. You want that air to be flowing through a HEPA filter, which you also want to replace.
So like COVID patients in hospitals are in like low pressure rooms. Would that be what they use? Because it's like they have a filter where air stays in the room, but like their air won't go out.
Okay, so yeah, they're probably using
they have a filter that goes through, like the air filters through.
Yeah, so that is probably a HEPA filter.
Oh.
Yeah.
I didn't think you knew that was the filter.
I don't know exactly what it is. I haven't read about it, but I can guess that it is most likely a HEPA filter.
Okay.
Yeah. The whole point is that it's very efficient at getting rid of very small things in the air. So we would want that in a COVID room. All right, so we know that visible light follows a certain wavelength, and things that are smaller than that, part of them are, or pardon me, part of the light spectrum that's smaller than visible light is UV light. So this can cause your thymine in DNA to attach to one another nearby. So instead of, take a look at this figure down here, instead of your adenine binding with thymine, so A and T together, it can cause two nearby thymines to attach to each other. So now we don't have our two DNA strands bound to each other. We have two thymines in what we call a dimer. So di meaning two. And that's going to be a problem for making a protein. So your polymerase is going to come in, and it's not going to be able to proceed past this dimer. Same thing with DNA replication. You mess with the DNA, you're going to mess with the entire cell. So this is what UV light can do. It can cause a thymine dimer. And then this next slide, we're going to talk about X-rays and gamma rays. These can break DNA, and that can cause the same problem. You have a break in your DNA. You can't make proteins. Without proteins, you're dead. If you can't replicate your chromosome, then you've failed to proceed into the next generation. So you will eventually die and not reproduce. So we're going to call X-rays and gamma rays ionizing radiation. They're going to cause ions. Remember, an ion is the gain or loss of an electron. What do we call the gain or loss of a neutron? Right. So if you don't want to heat something like your strawberries, for example, you can hit them with ionizing radiation to make them more safe to consume. I think you're fine going out into a field and just picking a strawberry to eat it, but that's on the small family farm type of situation. We're dealing with large industrial farms and human populations. We radiate things to make them safer for the general population. So that's foods, but then also heat-sensitive pharmaceuticals, so like your vitamins, certain drugs that we don't want to heat, you can hit them with radiation to make them safe to consume. Let's talk about some other ways to prevent food from being spoiled. So we can dry foods. We can make like jerkies or dried fruits. So if you take out the water, then microbes don't have that essential component for life that they need just like we need. Take out the water, they can't survive on it. Salting is commonly used in meat, so salt-cured porks, for example, or fish. So what this is going to do is, similarly to drying, you increase the solute concentration on the outside. Through osmosis, the water is going to move outside towards that gradient. So you've removed the water from the inside of the food. Microbes can't survive. The same is going to go for really sugary things like jams and jellies. Instead of salt, we have sugar. Let's see, yesterday we learned about temperature requirements for organisms. I think that was yesterday, maybe 2 days ago. So we know that organisms have their optimal temperature range. If you drop the temperature, microbes are going to grow more slowly, and your food will spoil less quickly. But you know that you can't leave food in the fridge for 10 months. Eventually, it's going to go bad. Certain organisms can survive. They're going to eat your food more slowly. Yeah, so with salting, we can put salt on the outside of a food, okay? If you put salt on the outside of something, there's more salt on the outside than on the inside. Follow me? Okay? So now water, through osmosis, moves from an area of, let me think about this. Water moves from a low solute environment to a high solute environment. So water wants to move and equalize the environment, right? It senses that there's less water outside, so I'm going to balance that out and move towards the high solute concentration. That's osmosis. So if you have salt on the outside, water is attracted to it, it evaporates, and then now you've removed water from inside of the meat, for example. So if their water is not inside the meat, then the microbes can't survive in it, basically eat it. So by putting salt on the outside of something, you're removing water from it without adding heat. Yeah, which is what you do with like drying. So a food dehydrator is going to keep air circulating at a low temperature for a long period of time. But salting you can do even in like a refrigerated environment. And then this is just a breakdown of everything we talked about. Definitely recommend studying this for your quiz. I'm going to ask you something about one of these. We spent 20 minutes talking about it. You should expect notes or to be asked something about it. But for your all's purposes, I think that the most important things to focus on are incineration, pressurized steam, and pasteurization. All right, so let's talk about some chemicals we can use to disinfect. So the book mentions iodine in a tincture form. A tincture is going to be an alcohol-based liquid. So we have 70% ethanol, I believe it was 2% iodine, and we can apply that to the skin to reduce pathogens before we give an injection or open the surface. So again, these are not sterilizing, but they are disinfecting. They are reducing the number of organisms, not killing all of them. 70% is generally what I've seen. We're going to talk about alcohols at the end. 70% is going to be the most effective concentration of alcohol. So if we use something to eliminate or reduce microbes on a living surface, like our skin, we're going to call it an antiseptic. So this bottle over here, this is not an antiseptic, but your mouthwashes are antiseptic. As opposed to a disinfectant, which is what this spray bottle over here contains. So disinfectants are used on non-living surfaces, tables, beds, floors. Antiseptics are used on living tissue. Anti, meaning removal of, septic is referring to life or bacteria. So we can say that sepsis is the condition when you have contamination from. All right, we're back. We are still recording. Share.
Thank you.
Enter. Yeah, you're welcome. Thanks for letting me know. All right, so sepsis, contamination from microbes. In your blood, sepsis refers to a blood infection. Asepsis, put an A in front of something, that is the lack of, so the lack of microbial contamination. Asepsis. So what I think is very important, you all hear this in lab, is aseptic technique. How do we keep us in our environment free of disease-causing organisms? That is called aseptic technique. You don't want to be giving it to your patients. You don't want to be coming home and giving it to your loved ones. So we will practice aseptic technique in here and in the lab. There we go, we're back. All right, and let's just keep with it. Here we go. All right, so an example of degerming, washing your hands see. So we are back now. All right, and y'all can see me. So things to focus on would be, so something like, hold on, let me share my screen. So something like this slide is worth your time. Can you all see my screen now? Yeah? Okay, cool. So this is a nice summary slide. You can see all these different moving parts that we talked about. So if you look through this and you can say, okay, I can explain dry heat, but I don't remember moist heat. Go back through and focus on that, because all the following slides explain this figure. Once we get to the end, we have this figure here. This is going to, again, just kind of break down everything that we talked about. So using these summary slides is a good way to, also like this is a good way to just kind of focus your studying. And then as you go through, you might say, wait, I'm less familiar with X-rays and gamma rays. And then you can go back in that slide or that part of the chapter to learn about that. So I think these summary slides are a good place to start, or rather, a good second step. Starting is always the textbook. Read the textbook, highlight out of the textbook, and then come back in here to these summary slides. That's what I would recommend.
I meant earlier you mentioned like pasteurization and then a couple more things, but like I was away from the screen for a second and I didn't hear that. But
Okay, yeah.
Important.
Yeah, so what I'm saying is, so I mentioned on this slide specifically that incineration, hold on, let's get pointer option, laser pointer. Out of this slide, incineration, you are going to use a flame in lab, that's important. Pressurized steam with an autoclave, also important for lab. And then pasteurization, just because the foods that we eat are pasteurized. So I'm saying that you all will see these in the future, so I think it's worth learning about it now, just for your education's sake. Things like UV, X-rays, gamma rays, they're important, otherwise I wouldn't make you learn about it. But what I'm saying here is that incineration, pressurized steam, and pasteurization are things you will see over and over again throughout the semester. All right, so now that we're back up and running, so I told the people in class to kind of like go take a little break. I'm gonna go see if I can pop out in the hallway and find them, and then we'll get rolling. All right, I can hear them coming back in. All right, let's get back at it. All right, so some stuff to consider. If we're gonna use a chemical agent to clean an environment, we wanna know that that agent will get rid of whatever microbe we're trying to get rid of, right? So organisms have different susceptibilities, and we want to target that susceptibility. Certain chemical agents are going to act at an optimal pH or an optimal temperature, so that's something to consider. Something might only function if it's at room temperature, but not function at an acidic pH and a low temperature. So that's something to consider. So this is the pH and the temp of your chemical agent, not the microbe. Generally speaking, higher concentrations of chemicals are going to be more effective, right? If you have more of something that's harmful, it's probably going to kill better. We talked about ethanol earlier. Ethanol is not like this. 70% ethanol is more effective than 95% ethanol. That's going to be because water is the other 30%. Ethanol is soluble in water, and we want that ethanol to go into the cells of bacteria. So the ethanol causes the membrane to be damaged, and then the water is going to transport the ethanol into the cell. So we've broken the membrane, and then the ethanol can go in and cause damage to proteins. Dysfunctional proteins cause irregular metabolism, cell dies. You've also ripped it open with ethanol. If you have more bacteria, more microbes, you're probably going to need to adjust your cleaning technique. Things in a biofilm are going to be harder to clean than organisms that are not in a biofilm. So cleaning of catheters is absolutely essential. We want to know about the environment. So are we using something in a closed setting, or is it in open air? We're not going to use a carcinogenic fume in a hospital room. And we want to know if microbes form endospores. If so, we want to target them specifically. What types of bacteria can form endospores? Gram-positive or negative? Positive. Gram-negatives are not producing endospores. Only some gram-positives. So if we're going to make some sort of a chemical that's going to kill bacteria, we want to know that it can kill or slow down the growth, but we don't want to hurt ourselves. So let's go back to Joseph Lister in the 1800s. He used phenol, carbolic acid. So that kept people from dying from amputation, but it's very caustic, so it damages the skin. But damaged skin is better than being dead, right? So we want to try and avoid hurting ourselves as we try and keep ourselves healthy and well, right? Generally, we want it to be soluble in either alcohol or water, easy to apply. We want it to last for a long time, so we probably have it to be refrigerated. We want it to be used in a diluted form, act quickly. We don't want it to stain ourselves, or let's say you're cleaning a table. You don't want to stain your table, right, with your disinfectant. You don't want to bleach your skin. And then also corrosive, we don't want it to burn a hole in our table, in our clothes, in our skin, whatever it is. Okay, generally, we want to use something that doesn't have a bad smell to it. I'm going to put a little evolutionary spin on this. If something smells bad, it's probably dangerous, right? So things that smell bad are probably harmful to you. Avoid things that smell bad. And then just with all things, we want it to be affordable. We want it to be accessible, inexpensive. I'd say 70% ethanol is probably the most universal, cheap thing you all are going to see in your healthcare settings. All right, so let's talk about how, yeah, question.
So like Paramedic stuff is like 70%, and like why do some doctors use isopropyl?
Why do some doctors use isopropyl alcohol as opposed to ethanol? I'm not sure why. I always use ethanol. Isopropyl alcohol might be cheaper. Ethanol is complicated because it makes us intoxicated. So we have to add things to it that'll make you sick if you drink it, so that people don't have to pay a liquor tax. So Walgreens can't just sell 70% ethanol with 30% water. Because then people are going to go and they're going to drink it and get drunk all day, and then those companies aren't paying a liquor tax. So that gets into like regulation, the Alcohol, Tobacco, Firearms Group, ATF, right? So like it gets into whole like regulation stuff. Isopropyl or ethanol are functional antiseptics. Either of them can be used. You'll find both of them in the little pads and in hand sanitizers. You can. But isopropyl alcohol will make you sick if you drink it. So it's probably just cheaper, honestly. That's probably why they're using it. Yeah. But you all know your mouthwashes like Listerine, it has 20% ethanol. So some hardcore alcoholics will drink Listerine and they'll get drunk, but then they'll also die because we have phenolic compounds in there. So don't drink alcohol in mouthwash, please. If your friends are doing it, tell them to stop. Okay, so how are we going to tell how effective a chemical is? One way is the phenol coefficient. So going all the way back to Joseph Lister and his phenol. Again, carbolic acid, phenol, same thing. So we're going back to that as a standard. The way this system works is if something is more effective than phenol, we're going to give it a number greater than 1. If it is less effective, it is smaller than 1. I doubt you all will ever do this. But yeah, we'll expose an organism to a compound, see if it grows relative to phenol, better or worse, and we'll give it a number. Not super important. The use dilution test, I could not find a figure anywhere on the internet about this. Your book doesn't have one. No, the last one. The phenol coefficient stuff, that's not super important. The use dilution test. So I couldn't find an image of this, but it's pretty straightforward. I've never done it. Basically, you're taking a cylinder of stainless steel. You're going to take a culture of bacteria, so in a broth, in a liquid, and you're going to put them on that stainless steel. You're going to let them dry. So now we have bacteria on steel. Okay? You're going to dip that into some fresh media. So, or pardon me, not into media, into whatever chemical you're exposing them to. So let's say bleach, ethanol, peroxide. So you have bacteria on steel, expose them to a chemical, then you're going to place that steel in fresh medium. You're going to see if it turns cloudy. What does it mean if it turns cloudy? Turbidity. So turbid is cloudy. Right. So what does that indicate? If we have turbid.
There's bacteria there.
Yes, there's bacteria there. Perfect. So if the liquid turns cloudy, that means that there's stuff in your liquid. That stuff is life, bacteria. So if it doesn't turn cloudy, then that'll tell you that whatever the chemical was killed the bacteria that were on the steel. That's how this plate or this test works. You all will almost definitely do this in your lab. I haven't seen the syllabus. The disk diffusion method is kind of similar. So you're going to take a petri dish. You're going to lay out what we're going to call a lawn of bacteria. So you spread bacteria across the whole plate. Then you're going to take these little paper discs. So these are absorbent paper circles. You dip it in, say, bleach or mouthwash, just water, whatever it is. So you're going to expose the paper to a chemical, and then you're going to place the paper on top of your freshly inoculated plate. Let it grow overnight, and then you're going to see how effective each of these are at killing the bacteria. Pointer option, laser pointer. So what we're going to do is we're going to measure what's called a zone of inhibition. So this bleach disc, for example, the bleach is going to kill organisms in this radius. So from the center of the circle, or from the edge of the paper, we can measure that radius and then compare it to the other discs. If you have a larger zone of inhibition, we're going to say that you are more effective at killing than discs that have a smaller zone of inhibition. Notice water doesn't kill these bacteria. Bleach is the most effective. So this is also something that I recommend studying. I will probably make some sort of a PowerPoint slide, or just find any sort of a stock image of this on the internet, show it to you and say, which of these is the most effective, which of these is the least effective. And so you'll look and you'll say, all right, well, water is the least effective, or hand sanitizer, they have no effect, whereas bleach is the most effective, followed by maybe alcohol and iodine. Does this make sense? Yeah, pretty straightforward. So if you ever get stuck in this whole class, any science class, just break it down according to the name. Disc diffusion. You have a little paper disc, the chemical diffuses out of it and will kill something. All right, so we have 3 halogens that the book mentions. These are going to make your enzymes less functional and impair protein synthesis. Things like fluoride are found in water and toothpaste. We didn't start adding fluoride to water until the 1950s. We started doing that because this one population was drinking well water, and they didn't have cavities. So somebody's like, well, what's in their water? They found a lot of fluoride, so then we started putting fluoride in all of the water in the US as a cavity-preventing method. That's why our water has fluoride, to help us keep our teeth. And, surprise, that's why we find it in our toothpaste. We're going to keep our enamel nice and strong. And it's also found in fungicides. Chlorine, this is valuable because it can kill spores. We can also use it to clean some wounds. I have not seen that in the past. And it's used in water treatment facilities as well.
I have seen chlorine and bleach used to clean open wounds.
You have. Do you remember what type of chlorine was used?
I do not, but I could probably figure it out for you.
Yeah, you know, if we could just touch base, maybe over email or later tomorrow, I'm genuinely curious about that.
Yeah, I could see if I can find out for you.
Yeah, that'd be great. And so you said, is that why we don't drink pool water?
Yeah, I went to
No, I mean, I feel like that's probably a good reason to not drink pool water, because we have to treat it with a lot of chlorine. Yeah. And then iodine, we've talked about this a couple times. As a tincture, this is used as an antiseptic. You all have seen this probably. It's kind of like that red color. Also, if you're into camping, hiking, you can get iodine tablets. And it's not the best way to do this. Like if you don't have one of these life straws that are out now, you can take pond water, lake water, put an iodine tablet in it, and it is more safe than if you didn't. So keep that in mind if you go camping. Yeah, what's up? Tincture is an alcohol-based solution. Yeah, so if you were to take, say, like vodka and basil leaves, you can make a basil tincture. Yeah. It's going to extract organic compounds. Let's test our memory retention. What's another name for phenol? Carbolic acid, that's it, right. So, phenol and phenolic compounds. We've talked about how phenol is caustic, so it's dangerous. It's also expensive, and it smells really bad. So, we know better than to use it now. We're going to use derivatives. So these are less toxic and more effective. So let's use these as, as opposed to phenol itself. Commonly, we're going to attach 2 phenolic compounds to one another you have two of them, we're going to call it a bisphenol. What is a bisphenol that you all have definitely heard of in plastics? BPA? You heard about that? Bisphenol A. It can just regulate hormones, we think, in humans. So a lot of your plastics will say BPA free. Now you know what a bisphenol is. All right, here we are. Let's share, going back. All right, we talked about phenol, triclosan. Go down. All right, so heavy metals are going to act on proteins of bacterial cells. So things like mercury, copper, silver. When we say heavy metal, that refers to its atomic mass. Atomic or molecular, I don't recall. Either way, it is a big molecule, big atom. So if they act on proteins, dysregulate metabolism, cell can't survive, it dies. So things like brass on a doorknob or a copper doorknob automatically disinfects itself. Think about that if you're building a house, right? Instead of plastic or wood, go for something like a heavy metal. In the past, silver, I think it's silver nitrate, silver nitrite, some sort of a silver compound is added to newborns' eyes to prevent eye infections. I don't know if that's still used. The book said that some sort of other antibiotic is used. Anybody with a family know if silver nitrate is still used? No? Okay, good. We figured out something safer.
I know that it's becoming more common for people to have allergies to silver and like silver and gold, so that might be why they don't use it anymore.
Okay, yeah. And it's like, do it until you recognize the problem, and then find a better solution, right? We've talked about alcohols quite a bit. So again, these are going to denature proteins. They're also going to disrupt cell membranes, so help the cytoplasm to just kind of rupture out of the cell. So you break open a cell with an alcohol. We talked about vegetative cells being susceptible to alcohol, but not spores. Spores can survive 20 years in alcohol exposure. They are not affected. All right, I think we have one more slide after this, maybe. Two more after this? All right. So soaps and detergents, we're going to call these surfactants. So it is something used to clean a surface. So a soap is going to grab bacteria on a surface. It's going to put them into little enclosed particles so that they wash away off of the surface. That's what a soap is going to do. So that's why you don't just apply soap, you have to put it on and then rub it around. So you have to literally wash off the things from your skin. Emulsification is using the fats to trap the water. So things like egg is an emulsifier. Dish soap is an emulsifier. So this is a simple experiment you all can do if you take water and vegetable oil and shake it up, they'll separate. If you put dish soap in there, you will have an emulsification. The dish soap will grab all the fats. Mayonnaise is emulsified with egg. All right, so a detergent is going to act a little bit differently. This is going to attack the cell membrane of microbes. So these are generally carrying a positive charge. So do we call it a cation or an anion? The cation, right. So cell membranes, keep in mind these have a phospholipid bilayer. That phosphate group carries a negative charge. The positive charge of the cationic detergent attaches to that and disrupts the cell membrane. There's a certain group of detergents that the book mentions called quaternary ammonium compounds. Shortened, they call them quats. And here are a couple examples here. So just be aware that these are detergents, and they're going to act by disrupting cell membranes. I don't need you to know specifics about the ammonium part. So we can use these to clean industrial equipment, as skin antiseptics, mouthwashes, or in contact lens cleaners. So I recommend, look at your active ingredients in all your household products. Your contact lens cleaners, your mouthwashes, your soaps. Review all this stuff and make use of your education. It's really valuable. Okay, peroxides. We've used hydrogen peroxide. It comes in that brown bottle. It bubbles up, right? We're going to learn about why it bubbles. So our tissues, as well as bacterial tissues, most bacteria are going to contain catalase. I say most. Some bacteria contain catalase. It is an enzyme that is going to convert hydrogen peroxide into water and oxygen gas. So this is commonly used on skin surface, so like scratches, little abrasions. But the text recommends not using it on deep open wounds, because it can be damaging to our tissues. But since it produces O2, so molecular oxygen, this is effective against anaerobic pathogens, because that oxygen is going to cause them to die. Keep in mind, though, again, our tissues, we make catalase, this enzyme. Some bacteria can also make catalase. It's a really simple test. You take bacteria, put them on a glass slide, put hydrogen peroxide on them. If they make catalase, you'll see it bubble. If they don't, it won't bubble. The bubble is the conversion of a liquid into a liquid and a gas. So that's what you see when you put hydrogen peroxide on your body. You've created a gas from a liquid. It's nice knowing how the world works, right? So we've talked a lot about disrupting protein use through metabolic pathways, disrupting cell membranes. Aldehydes are very reactive with nucleic acids or proteins. Things like formaldehyde, very reactive, very dangerous. We use this to fix dead people so that we can use them in a cadaver lab and they don't smell bad. But you can leave a fixed human out at room temp for months at a time, and there are no bacteria on them, so they don't decompose. Pretty cool. We can also use this, not formaldehyde specifically. I forget what, I think it was, so glutaraldehyde is what they're using. People in a healthcare setting are using to clean fiber optic endoscopes. So if you have some sort of a surgery where they put an endoscope in you, you want to make sure that's clean. You can use an aldehyde, but it forms a film, so then you've got to clean it with sterile water. It's a whole process, right? Ethylene oxide, this is a gas. It's explosive and it's carcinogenic. So I don't think we're going to want to use this too often. But if you have to sterilize something like wood, paper, leather, metal, rubber, that's where you'd use this. And so what I think about is, I purchase these goods. I'm using paper, I'm using leather, I'm using wood all the time. Some workers are exposed to these gases. Keep that in mind, right? These things don't just show up on store shelves. People are exposed to these deadly chemicals. All right, chlorine gas, this is less toxic, non-carcinogenic, and this was used, as they mentioned, to clean the anthrax-exposed male. You don't want to put it in an autoclave. You don't want to burn it, but you can expose it to a gas to get rid of the anthrax causing bacteria. This can also be used to pump through air ducts. We can clean entire facilities or hospital rooms with this gas. And again, we're going to use this as opposed to the explosive carcinogen ethylene oxide. And then this is just the summary slide of everything that we talked about. So what I recommend you do, we have 3 good summary slides here. We have this one. We have this one. And this one. So these are 3 summary slides. So I recommend going over these, looking at it, and try to either in your head or verbally talk about each of these. Just with this out here. Talk about why incineration is used, what's it effective against, what's it not effective against, and then use that as a way to gear your studying for quizzes in the exam. So that's the material. Let's talk about what we're going to do tomorrow. Tomorrow's chapter 10, we talk about antibiotics. We're going to take quiz 2 through the Respondus Lockdown browser. So please make sure you have that up and running. You're going to have to do that with your webcam running for tomorrow. I've had a lot of people reaching out to me recently about how to study, how to do well, what resources we have. And so far, I've been able to help out everybody who's asked for it. So if you're struggling, let me know, and I got you, okay? Questions about anything? No? People online, do you have questions about anything?
I had a question.
Yeah, may I hear it?
For the paper, when is that due? So that I can get a good idea of when to start on it.
All right, so the paper is going to be due at the end of week 4. I think that was January 7th. Thank you.
Okay.
So you're going to come back from break, and then it's going to be due that Thursday that you come back from break. That's January 7th.
Okay, thank you.
Yeah, you're welcome. All right, anybody else have questions?
No.
All right, cool. I'm going to go ahead and stop this recording and post our lecture. I'll probably have to post it to YouTube because for whatever reason, Canvas is full of memory. It's not letting me upload video files. I've never seen this before. So we're going to have another web link, whatever. You have access to it. Today's going to be in 2 parts because of our internet connection issues.